Hi everybody, welcome back. We just finished talking about the difference between observation and inferences, and we introduced the scientific method. So on this slide here, I have the 6 steps of the scientific method. You may want to take a moment and pause the video and go ahead and write these down. You are going to have to know all 6 of these steps in order, and this is how scientists are going to conduct their experiments. So they start by stating the problem, then they gather information, form a hypothesis, test a hypothesis, analyze data, and state a conclusion. And we'll go through the details on that here in a second. You see at the top of my slide here, I have the word experimental design. That is just a synonym for scientific method. The scientific method and experimental design are the same thing. So our first step of designing an experiment or a scientific method is stating the problem. And underneath that, I have a pretend experiment that we're going to talk about through the rest of this video. And my problem here is, what effect will sunlight have on the height of a rose bush? So that's the question that we're trying to answer using this experiment. After we have our problem, we're going to gather information, and that's through research and measurements. We're going to do things like measure the height of our different rose bushes, measure the amount of sunlight they're getting, so on and so forth. After we've done that, we are going to form a hypothesis. Now, a hypothesis is an educated guess, and that's going to state what you expect to happen. And it's going to state the relationship between those two variables that we're going to test through observations and experiments. So it's very important that when you're doing your experiments and when you're writing them, when you're writing up a lab report, that you write this as an if-then statement. So it's a complete sentence that begins with the word if, and then you say something like in this experiment, for example, we say if there is more sunlight, then there will be an increase in the height of the rose bush. So that's our hypothesis here. You can see we have if there, and then we have a then there. The hypothesis is going to be very similar to how you're going to title your experiment. And what we have here is the effects of sunlight on the height of the rose bush. Or you can remember that as the effects of IV on DV. And what IV and DV are is independent variable and dependent variable. So in our statement here, sunlight is our IV, the height of the rose bush is our DV, and we'll get into that more here in a second. So after we've formed our hypothesis, we're going to test it. So our experiments are going to use variables, controls, and constants. Very important that you know the definition of these 3 terms. We're going to go over them in a second. You may want to go ahead and copy those down. So variables. We have 2 different kinds of variables like you just saw. We have the IV and the DV. So our IV stands for independent variable. This is the manipulated variable. When we say manipulated, we mean that it's something that you purposefully change. An easy way to remember that is it's the thing I change. IV, I change it. So this is going to be the effect of sunlight on the height of the rose bush, right? So the sunlight is our independent variable. We're taking rose bushes out of the light and changing the amount they receive. So what happens is our IV affects our DV. Our DV is our dependent variable. Now that's the thing that changes in response to the independent variable. You may also see this written as responding variable from time to time. So these changes are something that are measurable quantitatively, meaning we can either measure its numbers or it's qualitatively observable. So our example, our dependent variable here is the height of the rose bush, and that's responding to the amount of sunlight that it's receiving. And then we have our control. It's important that you know that the control is the standard for comparing experimental effects. So what that means is we're going to have something that is normal and unchanged that we can compare the things we changed to. So no changes have been made, no alteration has been made to the independent variable. So in this experiment, what our control would be the rose bush that receives the normal amount of sunlight. So we'd just leave that rose bush outside in the sunlight, getting the normal amount of sun it gets for a day. Not all experiments have a control, but most of them do. So when we're doing this fourth step of testing the hypothesis, we're going to do repeated trials. So that's the number of times you do the experiment or the number of experimental repetitions. You have to repeat each different variable, each different independent variable. So for this one, we'd repeat the control, the rose bush that receives the normal amount of light. And then we'd repeat the other ones that we've changed, say our rose bush that's getting no sunlight or our rose bush that's getting light for 24 hours. We'd do it over and over again, and that's to ensure that our results are accurate. So it's important that we repeat our trials in order to have the most accurate results possible. One last term, and you may want to pause this and write this down. It's important that you know constants. Constants are all the factors in your experiment except for variables. These factors keep the same value throughout the experiment and cannot change in any of your trials. So, some examples of constants in this experiment are the amount of water. Each different plant is going to get the same amount of water each day. They'll each be planted in the same type of soil, the same type and size of pot, and they'll be kept in the same temperature. So constants are the thing that we do that are not changed. They have to be the same for each different variable throughout the experiment. So, while we're testing it, we're going to make observations. There are two different kinds of observations we're going to make. We're going to make qualitative observations, and we're going to make quantitative. So, qualitative right here, that's gathered using observations based on characteristics or descriptions, or you can think of as the qualities of the object. So, like the color, how it looks, so on and so forth. This kind of data is not something that can be measured or counted, so we're not going to get numbers, we're just going to get descriptions. So we have some examples down here, like the book is blue, the bacteria is round, and the surface is smooth. Our other kind of observation we're going to make is a quantitative observation, and that's an observation we get by counting or measuring things, or observing what you'd call the quantities. So basically, if you get a number for something, it's going to be a quantitative observation. So here are some examples of quantitative observations. There are 6 legs. The tree is 5 meters tall. There are 6 million bacteria. So let's take a look at this rock. This is a piece of granite. We're going to make qualitative observations about it. So some things we could say about it that are qualitative: it has different colors. There are crystals in it. That's what all those little specks are. It's small. This is not a very big rock. And then let's see, we're going to take a picture of a ladybug and we're going to do quantitative observations. So let's see. There is one ladybug. She has 5 spots, 2 wings, 6 legs, even though we can only see 4, and 2 antennae. And those are all quantitative because they all have numbers. So, we've finished our observations. We've finished the 4th step, testing our hypothesis. Now we are going to analyze data. So what you do is you're going to take all your data into consideration here. You're going to look at the good and the bad and start to form a pattern between that. After you've analyzed your data and looked at it, you're going to state a conclusion, and this is going to make an assumption based on the results of repeated trials. So when you state your conclusion, what you're going to do is you're going to refer back to your hypothesis, and you're going to say the hypothesis was or was not supported by the data because this. So if we hypothesized that the rose bush would grow higher with more sunlight, and it did, we would say our hypothesis was supported by the data because our rose bushes grew taller when they were exposed to more sunlight. So, a little bit more of vocab here that you're going to have to know. You've got a theory. You'll see this written as a scientific theory. This is a well-supported hypothesis, usually tested many, many times by scientists, but still up for debate. So that's something that can still be changed, but has been at least proven by data. So we have the theory of plate tectonics, the theory of evolution, things like that. Things like that can change, but they are supported by data and experiments. And then we have law, or a scientific law, which is something that's accepted as fact by scientists and society. An example of that is the law of gravity. This is not something that is going to change. Anyway, make sure you have these two things written down. That'll wrap it up here today, and we will finish up moving forward with some other stuff. Thanks, guys.